Hi everyone, my name is Silvia, and today I'm going to talk about the establishment of midbrain-hindbrain boundary. The division between the metencephalon and the mesencephalon is a fundamental one for the brain. This boundary is a major neuroanatomical division of the mature brain. The metencephalon gives rise to the cerebellum, while the mesencephalon gives rise to the midbrain, which includes the superior and inferior colliculi. During normal development, the region of the midbrain-hindbrain junction expresses the critical signaling factors that pattern the adjacent regions of the brain. To investigate whether the fate for these regions of the neural tube were restricted at this point of development, Alvarado Millar and the team transplanted a small piece of quail metencephalon to the forebrain of a chick embryo. As a result, cerebellum still developed from the metencephalon transplant. But in addition, the transplanted tissue had induced a new mesencephalon to develop from the adjacent forebrain neural tube cells. This experiment reveals the presence of a new signaling center, the isthmic organizer, which is reminiscent of the organizer transplant of Spearman, in that a small region of specialized tissue is able to repattern the surrounding regions when transplanted. Before we delve into the details of the signaling occurring exactly at the midbrain-hindbrain boundary, let's briefly go over how anterior-posterior regions are segmented in early development. Evidence suggests that Wen inhibition and Wen signaling set up the early anterior-posterior axis by activating two signaling molecules called OTX2 and GBX2. It is the boundary between these two signals that will ultimately give rise to the MHB. Then, what are the key signaling molecules in this midbrain-hindbrain boundary region? WNT1, Engrilled, and FGF8 were found to be the most important players in establishing the patterning here. In experiments, mice that were deficient in any one of these signaling molecules lost most of midbrain and cerebellum, which suggested that when Engrilled and FGF8 were functioning in an interconnected network, so deleting any one of them affects the expression of the other two. How can we put them all together in a bigger picture? The current model is as follows. IRX1 induces both OTX2 and GBX2 in a developing CNS. OTX2 and GBX2 actually cross-inhibit each other. And at the midpoint here, FGF8 is expressed, which can then activate Engrilled in those cells that express both IRX1 and OTX2. These cross-regulatory interactions set up and maintain the initial border through development. Here is another illustration to demonstrate the expression of some of the important signals. OTX2 in the anterior region and GBX2 in the posterior creates a sharp border between the midbrain and the hindbrain. This induces the secondary signaling molecules, which in this case are WNT1 and FGF8, and allows further cross-regulatory actions among the cells. Once the number of cells exceeds the range over which the signal can act, a new signaling source arises. This is the anterior neural ridge. At the anterior pole of the brain, where FGF8 once again acts as an important signaling molecule, this time inducing forebrain regionalization.